Well, let's go ahead and get started. And we're going to wrap up the discussion on metabolism today. And then we'll be done. And when we come back from break, we'll be done with this unit. When we come back from break, we'll be starting on bones and muscles, which is a whole lot more interesting. In fact, once we get done with this particular section on the metabolism, then we start moving into stuff that has real relevance to us as far as applications, considering, things like, how bones work and how our muscles work and stuff like that. So more than just stuff we can't see and hear. So, okay, let's move on. Anybody have any questions? Okay, then I'm going to go ahead and share screen and open this up. And everybody should be able to see the slide that says control of metabolic reactions. So let me go back a slide and we'll start here. Not in this one, let me go back. There we go, metabolism. What is metabolism? Metabolism is the sum of all chemical reactions taking place in our bodies. And so this is the reactions of building up energy, building up molecules and tearing molecules down and obtaining energy from the food that we eat. And that's what we're really focusing on right now, is the foods that we eat. The term cellular respiration is a part of metabolism. And cellular respiration isn't breathing. Cellular respiration is the process that we use to take the energy out of food that we eat and then use it in our bodies. When you see a calorie content on something like, a Coke is 220 calories, or an apple is 60, or a Snickers bar is 180, or something like that. Those calories represent the potential energy stored in the valence electrons. And all those molecules, those carbohydrates and fats and proteins linked together, the potential energy of all the valence electrons is that representation of 180 calories. That's what you could, that's the total amount of energy found in a candy bar, in a Snickers bar. And we want to take that energy and transfer it to our cells in our bodies. We want to eat that candy bar, and we want to take all that potential energy from those valence electrons and at least get some of it to use to pump our blood and to inflate our lungs and to generate enzymes and things like that. That's what we want. That's what cellular respiration does. We capture energy. Now, we know that our bodies depend on glucose as the source of the energy. Everything we eat gets converted to glucose, whether it's carbohydrates are broken down to glucose. If we run out of carbohydrates, we'll start breaking down fats and convert them to glucose. And we'll do the same thing to proteins. But once we get to glucose, we can't take the energy directly from the glucose all at once and apply it to our cells. It would be a really bad experience if we did something like that. We can't handle all that energy being released at one time. It'd be like if all the gas in your gas tank suddenly decided to explode at once. That'd be very exciting. Very briefly, but very exciting. You release all the energy in there. We don't want that. We don't want that going on in our cars. We don't want that going on in our bodies from all the food, all the energy that's in the food. So we do it slowly. We do it in a step-by-step process. This gets back to the whole point about empty calories. Empty calories are very high in sugars, high in carbohydrates. Empty calories release a lot of that sugar energy all at once. And so you get what we call a sugar rush. You know, because many of those calories that are in a Snickers bar are either going to be sucrose or already at glucose. And so we're going to have all this sugar to release at one time. We'll get a surge of energy and then we'll crash afterwards. It's where we get into the discussion about the empty calories versus the complex carbohydrates. It takes longer to break an apple down because apples are made of starch. It takes longer to break that starch down into glucose than it does to break sucrose or even corn syrup, which is a glucose type of sugar. It takes much longer to break down a complex carb full of starches than it does just to take a candy bar apart. And so we don't get all of our energy released at once from an apple. It takes time to release all the energy in the apple. We'll get all the energy, but we won't get it all at once. We get it a lot faster from a Snickers bar. And this is the difference between empty calories and complex calories. Empty calories give us the energy at once, but they have no lasting power. They don't stick around. You know, you have a surge and then you're done. And you wasted a lot of that energy. The complex carbohydrates give you the energy, but they give it to you incrementally so we can continue to use it over time. But regardless of how we get it, we still have to find a way to deliver it to the cells. And that's where this compound called ATP comes in. ATP, adenosine triphosphate. It's characterized by having a 3-phosphate structure on it. You know, ATP, the molecule ends with 1 phosphate attached to a 2nd phosphate attached to a 3rd phosphate. This 3rd phosphate, the number 3 phosphate, is held to the first 2 by what's called a high energy bond. Right there. We can break that bond and release energy to the cells where they need it. The ATP will bind to a location in the cell and it will break the bond for the 3rd phosphate and release energy. Now, the nice thing about the ATP is when you do that, when you break that high energy bond, when you get rid of this high energy bond here, you end up, let me clear all this off. when you break that high energy bond, what you end up with is ADP. It has two phosphates on its end, and there is what we call inorganic phosphate floating around. We haven't destroyed the phosphate. We haven't destroyed the ADP, the adenosine diphosphate. As soon as we get some more energy, we can shove this back together here. More energy allows us to make ATP again. So we're simply moving the phosphates back and forth. If we need to give energy to a particular part of a cell, we can send ATP to it. The bond will break between the second and third phosphates. We release energy, and now we've got ADP, a low energy compound, and inorganic phosphate. And if we have more energy, we'll put it back together again, and we can use it again and again. The problem with this is that ATP only lasts for 7 seconds. You make it, and in 7 seconds, we have to make it again and make it again. So it's not like we can store it. But what we do is we use the phosphate as a way to deliver our energy to the cells, a controlled way of delivering the energy to the cells. A molecule of glucose has about 680 kilocalories of energy. That's a lot of energy. A molecule of glucose has 680 kilocalories of energy. Each ATP is about 7.2 kilocalories. And it's a whole lot better to release it in 7.2 increments, one after another, than to release the entire mass at one time. So we're shifting, we deliver the energy using the phosphates. And whether we're, however the source of the energy is, whether it's from carbohydrates or lipids or proteins, the goal is to continue to make ATP, because we need to keep making it all the time, because we only get 7 seconds of it. Each molecule only lasts 7 seconds. Once we transfer the third phosphate to, like the sodium-potassium pump. Remember the sodium-potassium pump? We talked about that. We're pumping sodiums out of the cell and pumping potassiums back in. The sodium-potassium pump gets its energy from that third phosphate. So being able to contract our skeletal muscles gives us the energy for that, is transferring the third phosphate to the proteins in our muscle cells so that our muscles can have the power to contract. Okay, now, we use enzymes to make this work. We use a variety of enzymes to make this whole thing work. The process of moving moving the phosphates around. Now, we know what an enzyme does. An enzyme has what we call an active site. The active site is designed to have certain molecules land on it, and only certain molecules. You know, if we call those molecules the substrates. Enzymes are what we say substrate specific. There's an enzyme for just about every type of chemical reaction we could think of. And the enzyme, for example, in our mouth called amylase, which breaks down starch, only works in our mouth. It only works on starch. It won't work on fats or proteins, and it won't work in our stomach. It only works in our mouth, because the amylase is designed just to have carbohydrates land on it, like starch. And that's the only thing that will fit there. Nothing else will fit on the amylase. And since amylase is a typical enzyme, it is substrate specific. The active site on the enzyme where the chemical lands, where the starch lands, is designed just to receive starch. And we always call the molecule that lands, we call those the substrates, and they always land on the active sites. Anyway, now. So in our bodies, we use a type of phosphorylation, big term here, phosphorylation. It means phosphorylation means we're going to transfer the phosphates around. And we can transfer the phosphate from the ATP to the location where we want to do something, or we can make ATP by transferring, if we have energy from glucose, we can take a phosphate and put it back on an ATP and make the high energy ATP ready to go for 7 seconds. Or we can strip the phosphate off and go tell it to go out and do some work for 7 seconds. So anyway, the goal of metabolism of food is to produce ATP. That's where the energy is coming from, that we're using is from the ATP. Granted, it's coming from glucose, but the glucose energy is being passed to the ATP, and that's what we use to get stuff done. So if we are going to eat things, which we do, we love to eat food. There's 3 stages here that we have to pay attention to. We digest food. We start the process of digestion in our mouth. We continue it in our stomach, and we wrap it up in the first 10 inches of our small intestine. In the next 19 feet or so of small intestine, we absorb all the nutrients, the fats, the proteins, the carbohydrates get absorbed in the small intestine and go to the liver to be cleaned up, and then are sent out to the tissues. Once we get to the cells, now we can start working on what we've got. And we can put fats back together again. We can make proteins in the cell. We can make glycogen in the cell. We can store excess sugar in the form of glycogen. Or we can break down the glucose that we've got and release energy. There's a whole lot of things we can do inside the cell. We can use the energy to make proteins. We can use the available energy to put lipids back together again, or we can just store excess sugars. And that's not a bad payoff of energy. We get a little energy here in this second step. In the cytoplasm, we generate about 4 ATPs, or it says cellular processing in the cytoplasm. We get about 4 ATPs out of that. So that's not bad. We can survive off of that. But remember, in the cytoplasm, we're busy also making proteins, making large molecules, breaking down large molecules. We're going out and eating our friends and neighbors when they step out of line. All these things are going on in the cytoplasm. We're making antibodies. We are detoxifying chemicals. All this is taking place. So we don't have time to do a lot of extra work to produce ATP. But the last step, the oxidative step, takes place in the mitochondria. We call it oxidative because here's where we use oxygen. We haven't used any oxygen to this point. You know, we're breathing in oxygen. We haven't used any oxygen yet. In the mitochondria, we actually oxidize the glucose. It's like burning it. And when we oxidize it, we release electrons. And we break covalent bonds and we release electrons, and that releases a whole lot of energy. And then we can take that energy and produce a lot of ATP with it. In fact, we probably make 9 times as much ATP in the mitochondria as we do in the cytoplasm. You know, so because in the cytoplasm, we're going to obtain about 30 kilocalories of energy, breaking down glucose through glycolysis. We're going to get about 30 kilocalories of energy. But in the mitochondria, we're going to get about 260 kilocalories of energy. That's a lot more energy than we were getting before. So, you know, if you're visual, this is a nice little chart. It looks busy, but it's not too bad. Digestion at the top, proteins, carbs, and fats. Proteins are broken down to amino acids, and we take those amino acids in the cytoplasm and we build them back up into new proteins for us. Carbohydrates are broken down into glucose, and if we have too much glucose, we convert it to glycogen. And if we have even more than that, we convert it to fats. And then fats are broken down in our small intestine to glycerol and fatty acids for absorption, because the triglycerides are too large to absorb. So we break them down into glycerol and fatty acids, and then we reassemble them in the lymphatic system as fats. And then we do whatever we're going to do with that. It is the glucose that's going to continue first off, because glucose is what we use for energy. Glucose, when we get into the cytoplasm, will undergo glycolysis. Glycolysis, the brown box up there, and the end result of glycolysis is a couple of ATPs and the formation of something called pyruvic acid, and that leads us into the last step. This is the step with the big payoff, because here we have a process known as the Krebs cycle. Now, the Krebs cycle is not the big payoff. The Krebs cycle is leading to the payoff. The Krebs cycle involves preparing, the Krebs cycle strips electrons off of the glucose. That's oxidation. Oxidation is stripping electrons. Whenever you burn something, that's oxidation. You release electrons, you release energy, you release heat, you release light sometimes. If I were to this is oxidation. We see right here, I got the flame burning. The lighter is consuming the fuel inside here in the presence of oxygen, and I've got heat and light coming off. The reason that that's doing that is because I'm breaking the covalent bonds holding the fuel together. I think it's probably butane. And so we're breaking those bonds and releasing electrons. And when we release electrons, we're releasing potential energy. So we get heat and we get light from that. We're also giving off carbon dioxide and water. Now, we don't get a lot of light coming off of our bodies, but we do get a lot of heat coming off when we are capturing energy and using energy. So, but every time the point that was made here was that this is an oxidative process in stage 3. Oxidation strips off electrons, and so we release energy. When we release electrons, we release energy. So what the third step here, the Krebs cycle breaks electrons off of glucose, and then that goes into another area called the electron transport chain. And it sounds very complicated, but it really is pretty simple. The electrons then are used to power the production of ATP. So let's go on here, and this will make more sense as we go along. So, energy. We use all sorts of energy in our bodies. We use heat energy to warm up. We give off heat energy just from the breakdown of carbohydrates. We use light energy, so we can see light energy, light rays going into our retina. Sound, we hear things. We make noises ourselves. And so those are forms of energy. Those are pressure waves moving through the air. Electrical, we are electrically charged. We depend on nerve impulses to travel throughout our bodies. Remember the term action potentials. Action potentials are electrical charges throughout our body. Mechanical energy, we have mechanoreceptors in our body that respond to pressure and touch and itch. And we also respond to vibrations. We can hear things because the sound waves, the pressure waves hit our tympanic membrane, and that vibrates, and that's mechanical energy. And of course, there's chemical energy from the food that we eat. We're always changing these different types of energy from one form to another. Light rays that strike our retina are converted to electrical energy so we can send nerve impulses to our brain. Mechanical energy is converted to electrical energy so we can tell our brain what's going on. The same with heat. If we are cold, we shiver. We convert mechanical energy to heat energy. We sweat when we get too hot. So we're converting heat energy into chemical energy to do that. So we're moving things around all the time. The only thing wrong with this is that every time we change energy from one form to another, we lose a little bit of it in the form that we can't process. You know, we eat a Snickers bar and we get 180 calories of energy, but about 60% of that energy goes off as body heat. We can't help that. We always lose about 60% of all the energy we take in as body heat, but we get 40% to use. So that means we're about 40% efficient, which is pretty good. Now, chemical energy is what we're aiming for. We want to get everything to glucose, because glucose is what our bodies are designed to break down. We oxidize glucose to power our metabolism. And the process of cellular respiration takes the chemical energy in the foods that we eat have taken in, and we strip off electrons and we release that potential energy. So the 180 calories in a Snickers bar or the 60 calories in an apple becomes available to us to do things. You know, granted, we still lose 60% of it as heat, but the other 40% we get to use. If we're looking at cellular respiration, we have 3 steps that we go through. The first is called glycolysis. It takes place in the cytoplasm and only in the cytoplasm. We have the citric acid cycle, which is another name for the Krebs cycle. Krebs cycle. You know, he must have lost an argument at some point with some international union on chemistry because they took his name off of it and called it the citric acid cycle. But it really is the Krebs cycle because he's the biochemist that discovered it. You know, probably didn't have much of a life either, but that's a whole other story. So anyway, Krebs cycle. The Krebs cycle takes place in the mitochondria. Electron transport takes place in the mitochondria. Remember, every biology textbook will always call the mitochondria the power plant of the cell. You know, well, it's more like the power converter of the cell, but you know, we'll let it go as the power plant of the cell. So the Krebs cycle and electron transport take place inside the mitochondria. Glycolysis is taking place inside the cytoplasm. The end result always of cellular respiration is going to be the release of carbon dioxide, the release of water, the release of ATP, chemical energy in the form of ATP, and the release of heat. You know, we have carbon dioxide comes from the Krebs cycle. It's the only place in our bodies where we generate carbon dioxide. Water comes from electron transport. ATP comes from all 3 of these, and heat, 60% of the energy available goes off as heat. Our skeletal muscles are why we maintain a constant body temperature. The amount of heat coming off of our skeletal muscles when they use up so much glucose maintains our body temperature at 37 degrees Celsius or 98.6 Fahrenheit. So Both of these process, the process of glycolysis and then Krebs cycle and electron transport involve both anaerobic reactions that don't use oxygen. Glycolysis doesn't use oxygen. Glycolysis doesn't need oxygen. It is an anaerobic process. But the downside is it doesn't give you very much ATP. Glycolysis gives you 4 ATP. The Krebs cycle, which is also anaerobic, gives you 2 ATP. The aerobic reactions that use oxygen give you the most ATP. The electron transport chain uses oxygen. That's where oxygen ends up getting to when we are breaking down foods. That's where the role of oxygen comes into play. Now, to understand this a little better, this whole process of gaining and losing electrons is they're called oxidation-reduction reactions. Chemists get all excited about oxidation-reduction reactions. If a substance is oxidized, it loses electrons and loses energy. And oxidation can be fast, it can be slow. Consider if you ever knew of anyone, or you did it yourself, left a shovel or some sort of metal garden tool outside over the winter. What happens? What's it look like in the spring? It's rusted, right? Okay, it's covered with rust. Rust is oxidation of iron. It's a very slow oxidation, but it really is no different than what happens when you eat a candy bar or an apple. The only difference between the rusting on a shovel and breaking down an apple is the speed. But you're still oxidizing the material. But in the case of the shovel loses electrons and gets weaker over time till you get the shovel. You find, oh, look, I left this out all winter, and you go to use it for the first time and it snaps in half because you've lost, you've taken so much energy out of the shovel itself. It happens. That's why we have to keep buying new shovels, I guess. I don't know. When anything is oxidized, it's going to give off electrons and give off energy. When you're burning gasoline in your engine, you're oxidizing the engine. The gasoline, it's releasing its stored energy, its potential energy, and all of its valence electrons is being released. And you can use that to power your car, or the Snickers bar, or the apple, or the potato that you're eating. You're stripping you're breaking the valence electron, you're breaking the covalent bonds in the valence shells, and you're releasing those electrons to run free and release energy. The only problem is that we don't want those electrons to run off by themselves and just randomly disappear. There always has to be another molecule there to grab them. So when we are breaking the the covalent bonds, releasing the valence electrons in gasoline or in iron or in food, those electrons are going to be released, but they have to have somewhere to go. And this is where oxygen comes in. Good old oxygen. Remember oxygen? Here's an oxygen atom right here. It has an atomic number of 8. That means it has 2 electrons in its first shell, and it has 6 electrons in its second shell, and it has room for 2 more. Oxygen loves to grab electrons because oxygen wants to be stable. Oxygen doesn't think it's stable because it only has 6 electrons in its outer shell. It has room for 2 more. It wants to gain 2 more. It'll do anything it can to gain those extra 2 electrons. So whenever something is going to get oxidized, oxygen has to be there for it to occur, because oxygen is going to grab the electrons coming off and get stable. Well, the trick with oxygen is that when you grab an electron, electrons always travel with their BFFs. In this case, an electron always travels with a hydrogen ion, always, because that's a balanced mixture. The negative charge of the electron is balanced by the positive charge of the hydrogen. So the hydrogens and the electrons always travel together. So what happens when oxygen grabs an electron? It gets a hydrogen with it. So it picks up an extra electron here, an extra electron here, and ends up grabbing hydrogen. And guess what you get? H2O, water. Whenever we oxidize something, the end result's always going to be the production of water. Oxygen grabs electrons that are being released from their valence shells. Because oxygen really wants to get those electrons to itself. The electrons are coming along with their hydrogen ions attached to them. And all of a sudden, yes, oxygen's now happy because it has picked up 2 more electrons in its outer shell, but it has 2 hydrogens with it. And so we're sharing electrons there. Hydrogen's happy, oxygen's happy, and we've got water molecules, and we've released energy that we can use. So this is why oxygen is so essential to us. Without oxygen, without oxygen, we can't release those electrons. We can't release the electrons in food. We can't release very many of them anyway. So we say that an oxidized substance gives off electrons and gives off energy. The substance that captures the electrons is reduced. It's reduced. It gains electrons and theoretically gains energy. So when we're making water, by adding hydrogens and electrons to the oxygen, we are gaining electrons and theoretically, and we're gaining energy because we're gaining the potential energy of those electrons in the valence shell of the hydrogen and the oxygen. We call these redox reactions. And just to confuse everybody, the donor, the atom that gives up the electron, is called the reducing agent, and the electron acceptor is called the oxidizing agent. Well, oxygen always accepts electrons. So oxygen is always going to be an oxidizing agent. It's not the only one that accepts electrons, but it is always, it is the most common oxidizing agent. If you have a metal object, like a shovel or something that you have left outside all winter and it's still in pretty good shape, if you paint it with a substance called Rust-Oleum, it seals the surface and oxygen can't get in anymore. No more rusting, at least till the paint wears off. So anyway, the substance that accepts the electrons is the oxidizing agent, the oxidizer, if you will. Oxygen is an oxidizing agent. The donor is the reducing agent, because the donor is going to give up electrons, and whoever grabs them is going to become reduced. They're going to have more negative charges than they did before. There we go. Okay, now, and this confusing little diagram here is trying to show you what's going on. If you consider that the circle with the B in it is oxygen, oxygen is always going to become reduced when it grabs electrons. This compound up here compound A has electrons, and those electrons get stripped off, and it's oxidized. We've released electrons. We've given off energy. Here we have compound B, which will grab the electrons. It's reduced, and the chemists say it's reduced because there are now more negative charges on it than there were before. That's chemist talk. So, yeah, it says reduction is called that because adding the electrons reduces the number of positive charges. It means there's more negative charges, more electrons. So anytime anything is burnt, anytime anything is chemically destroyed like this, that's oxidation. Yeah. Burning the candle is oxidation. So this is oxidation reduction in action. We give off carbon dioxide and we give off water. Now, to demonstrate this for you, and I've spared absolutely no expense on this. This is, here we have a candle right here. And if I light the candle, it's a real candle. You can see the flame on the candle. It is going out. There we go. Okay. See it, there we go. You can see it a little better now. See, bring it in just a little bit. That doesn't help. Camera doesn't seem to like the flame. Okay, there you can see the flame on that. So, candle is burning here. We've got a flame. Now, it's burning because there is oxygen available. Now, if I take this large beaker and place this over the candle, don't break it. What's going to happen? What's going to happen? Well, I think we all expect the candle to go out. Anytime now. As long as there's oxygen available, the candle will continue to burn. But you notice that the wick seems to be getting smaller, and the flame seems to be getting smaller and smaller here. There we go. And it's gone. Now, if you take a look at the side of the beaker, you can see a very thin layer of condensed water vapor on here. There's a little, it is somewhat cloudy on the inside. If I were to take my finger and rub it across here, you could see, I guess it evaporated immediately. I'll show it to you again. What's happening here is that the products of oxidation are always going to be the release of energy with electrons. Also, you're going to have heat and or light and or heat, but you're also going to give off carbon dioxide and you're going to give off water. Because the electrons that are being released with their hydrogens. The oxygen in the air inside the beaker is grabbing those electrons and hydrogens, and it's going to condense as water vapor on the side, on the side of the beaker. And pretty soon it's not going to be as clear as it was, and it's going to start getting cloudier as the candle goes out. I think you can sort of see on the sides there where you can see the condensation on the inside of the beaker, particularly on the edges of it there. Now, why did the candle go out? Well, the typical answer would be to say it went out because of it used up all the oxygen, but that's not true. The reason that the candle went out is because carbon dioxide was generated and displaced the oxygen around the flame. If the beaker allows the accumulation of carbon dioxide, pushes the oxygen out through the opening at the bottom of the beaker, and the flame goes out. Not because it used up all the oxygen. There was plenty of oxygen inside there, but the carbon dioxide, which is heavier than atmospheric oxygen, carbon dioxide is heavier than atmospheric oxygen, and it settles down on top of the flame, pushes the candle out. So we know that carbon dioxide was being formed because the candle went out. We know that water vapor was formed because we could see the condensation on the side of the beaker. And this is what we always get when we have oxidation. We use oxygen. When we are having an oxidation reaction, we are releasing energy, we're releasing electrons, we form water, and we also form carbon dioxide. Now, we do this in a very controlled manner in our bodies. We don't transfer those electrons all at once. Remember, the electrons are bringing along with their hydrogens. We don't transfer all of them at once. We transfer them in a step-by-step process. We do it gradually. If we did it all at once, we'd have something that looks like this. This is uncontrolled oxidation. The Hindenburg, which was a German airship, a dirigible, this is in 1937. The Hindenburg had massive balloons, silk balloons, probably bigger than this room, filled with hydrogen gas. Hydrogen gas, elemental hydrogen, is very explosive. And it doesn't need, once it gets exposed to oxygen, it becomes very, very dangerous. And all it would take would be a small leak in one of those big silk balloons they had for the elemental hydrogen to leak out, combined with oxygen explosively like this. This is uncontrolled oxidation. There was no way to stop this. Once it started, it burned out, and you'll notice that this is a real-time video of it. It burned out very quickly once the hydrogen was gone, and the explosion just, well, you see how it happened. It actually happened like you're seeing here in the video. And there were 100 people on the Hindenburg, and about 60 of them got out because it hit the ground fairly quickly after it exploded, and they just walked out. So, but this is the very rapid formation of water vapor and carbon dioxide, and releasing a whole boatload of electrons at one time. This is uncontrolled. This is theoretically what would happen to us if we try to release all the energy in a molecule of glucose at one time. Fortunately, we don't explode, at least not that I'm aware of. Now, in the part of our bodies where we don't use oxygen, there's two parts here to cellular respiration. There's the anaerobic part and the aerobic part. The aerobic part, using oxygen, gives us lots of a good payoff. The anaerobic part doesn't use oxygen, but it does use other oxidizing agents. We don't get a lot of payoff from it, but we're not looking to make ATP from this part. We use a couple of coenzymes, and the coenzymes that our bodies use, that we use, are NAD+ and FAD. NAD+ has a positive charge on it, and it will accept 2 electrons. It'll take 2 electrons and 2 hydrogens. FAD will take 2 electrons and 2 hydrogens. These are reducing agents. These are oxidizing agents that will become reduced, stripping electrons off of glucose. Now, the whole idea of cellular respiration at this point initially is to make some ATP. Overall, it's to strip off the electrons. Let's go on now. The aerobic cellular respiration is where most of the work happens. And this is what aerobic cellular respiration takes place in the electron transport chain. However, we do a fair amount of work ahead of time in glycolysis in the cytoplasm. Again, the whole idea is to get these electrons off of the covalent bonds because they have the most potential energy. So let's see. Now, the formula for cellular respiration is this: C6H12O6, that's glucose, in the presence of 6 oxygens, will give you 6 carbon dioxides. It will give you 6 waters. We saw that with on the beaker. And it will cause the release of energy, which we can capture as ATP. You know, when we're burning our candle, we're not capturing any energy. We're getting heat and light coming off of it. But if there was a way to capture that, we would. But this is the formula that cellular respiration will follow. And the thing to remember is the NADs are electron acceptors. They are the oxidizing agent. They will take they can pick up 2 electrons and 2 hydrogens, both of which become very, very important to us. So let's move here into electrons always take hydrogens. Yes, I said that. So. Now, all we're going to do is we're going to transfer the energy from food into electron acceptors and use that to make ATP. The electrons, we have high energy electrons. There's potential energy. There's electrons in the valence shells have lots of potential energy, and we're going to take that energy away from them and use it to produce ATP. So just how do we make ATP? Well, something to remember with ATP is each ATP molecule is 7.3 kilocalories of energy. So you get 7.3 kilocalories of energy for each ATP. The total amount in a glucose molecule is about 680 kilocalories. So you need a lot of ATPs to get energy off of this, to get a fair amount of energy. Now there's 2 mechanisms that we use. Substrate level, remember this, substrate level in glycolysis in the cytoplasm of the cell. Substrate level phosphorylation, that's glycolysis. That's where you're taking the enzyme and you're using, here's your enzyme right here with your active site and your active site. And the substrates are the molecules that land here. One of the molecules has a phosphate on it and the other one doesn't, and you bring them together. That's substrate level phosphorylation. You're either stripping off a phosphate or adding a phosphate. That takes place using the enzymes. Oxidative phosphorylation takes place in electron transport. Try to remember both of those statements. Substrate level takes place in the cytoplasm during glycolysis. Oxidative phosphorylation is taking place in electron transport. I guarantee you those statements will come back to haunt you later on if you don't try to commit them to memory. This is substrate level phosphorylation. Takes place in the cytoplasm. It's the first part of cellular respiration in what we call glycolysis. You have your enzyme, you have your substrates in place. You have the 2 molecules land there on the active site, and you transfer the phosphate from one to the other. Substrate level phosphorylation. Glycolysis uses this. Oxidative phosphorylation is in the mitochondria in the electron transport chain, and we use both the hydrogens and the electrons. Let me show you how this is done. Okay, so the first step, metabolism of carbohydrates. We're going to give off, in the presence of 6 oxygens, we're going to give off 6 waters, 6 carbon dioxides, and about 36 ATP, about 260 kilocalories of energy. And we're going to give off heat. The other 60% of the energy in a molecule of glucose comes off as body heat. But we're going to get about 36 ATP altogether that we can do things with. Now we have 3 ways to capture electrons, to capture energy. The first step is glycolysis, takes place in the cytoplasm. Krebs cycle takes place in the mitochondria. Electron transport and oxidative phosphorylation takes place in the mitochondria. So, and here's a simple little picture if you're really into visual pictures of stuff. This is a good review slide to go back to, but it's too busy to talk about right here. So, glycolysis takes place in the cytoplasm. It uses substrate level phosphorylation. It does not require oxygen. It produces 2 pyruvic acid molecules and one more. So far you have to try to remember all of this. We got one more bullet here. ATP produces 4 total ATP. And it produces NADH, 2 NADHs are formed. This is everything you need to remember about glycolysis. Takes place in the cytoplasm. It's substrate level phosphorylation. Does not require oxygen. It produces 2 pyruvic acid molecules. It gives you 4 ATP and it gives you 2 NADHs. Now, pyruvic acid looks like this. Here's your 6-carbon glucose. 1, 2, 3, 4, 5, 6. At the end of glycolysis, the 6-carbon glucose is split into 2 3-carbon pyruvic acids. That's where the pyruvic acid comes from. So let's clear this and the final results of glycolysis are this: 2 pyruvic acid molecules. They go into the Krebs cycle. If your body is experiencing something known as oxygen debt, and this occurs when you've been exercising vigorously. See, our bodies store oxygen in the myoglobin in our skeletal muscles. That's what makes the skeletal. That's what makes our muscles red. The presence of oxygen in the myoglobin. Myoglobin is sort of like the first cousin of hemoglobin, but it holds onto its oxygen so that the skeletal muscle cells can oxidize glucose. Lots of oxygen stored in our muscles. When we deplete that oxygen, which we can if we're, let's say we're, on a strenuous hike or working all day doing construction and we're really, really tired at the end of the day, what will happen is we have depleted all of the oxygen stored in our muscles. And our muscles hurt because of this. Because when we experience this phenomenon called oxygen debt, and it takes about 24 hours to reload all the oxygen into the myoglobin. If we've done that, if we've used up all the oxygen that's available, we're in oxygen debt. We're not going to make pyruvic acid. We're going to make what's known as lactic acid. And lactic acid accumulates in our muscles and makes our muscles very sore. Lactic acid accumulation occurs during oxygen debt, and our muscles hurt later. Strenuous activity, long-term strenuous activity causes our muscles to hurt because we're not making pyruvic acid, we're making lactic acid. Anyway, we're also going to produce 2 NADHs. The most important thing about those 2 NADHs is that they're carrying 2 high energy electrons each. So your NADH and ADH has 2 high energy electrons, each molecule. So you've picked up 4 high energy electrons here. You've made 4 ATP also, you know, and you say, oh, I made some ATP, you know, because you don't recognize we never recognize the importance of the high energy electrons. The downside of making this process, to get the process started, to get everything rolling, to oxidize the glucose initially, to make the pyruvic acid, it takes energy. We have to start it. We have to turn the key. It's like turning the key or pressing the button in your car, and we start the car. We use energy from the battery to start the car. So here we have to spend some ATP. We actually burn 2 ATP to start the process. We make 4 ATP, but we burned 2 to start. So we end up with a net gain of only 2, because we have to use 2 to start it. But we still end up with each NADH has 2 high energy electrons. And that's more important than the fact that we've made 4 ATP for us. So glycolysis, we make 2 pyruvic acids, we make 2 NADHs, and we have a net of 2 ATPs. Now, test-wise on this, if the question asked you for total ATP for glycolysis, the answer is 4. If it asks you for the net number of ATP, the answer is 2. Important thing to keep straight. Keep straight between total and net. Okay, now let's move on. Cellular respiration then is ready to go. We are ready to start the process of cellular respiration by moving the pyruvic acid into the mitochondria. Because we want to make more than just 4 ATP. We want to have a lot of ATP so we can go do things. We want to be able to contract our muscles. We want to be able to breathe. We want to be able to pump sodiums out of our cells. We want to do things. So we need to get more ATP. We want ADP to undergo phosphorylation, to have the 3rd phosphate put onto it in a high energy bond. Whether we're making it through substrate level or whether we're making it through oxidative phosphorylation, we want to be taking ADPs and jamming that 3rd phosphate on. So we can send that ATP out for 7 seconds to do things. This is what ATP looks like. There's the 3 phosphates, 1, 2, and 3. When we give up the phosphate, it drops back to its low energy ADP mode. If we have more energy available, well, first of all, we break the bond, release the phosphate. And now if we have energy available, we can take the ADP and the phosphate and put it back together again. And so for another 7 seconds, we have 7.3 kilocalories of energy available to do something. So we move into the mitochondria. we move into the mitochondria now. The pyruvic acid, the 3-carbon compound that was the end result of glycolysis, both of those pyruvic acids move into the mitochondria, usually one after another. They go into the mitochondria, and as soon as they get into the mitochondria, each one of those pyruvates gets oxidized. The pyruvates go in and they get oxidized, and we strip off some more electrons. NAD plus becomes NADH inside the mitochondria, stripping off more high energy electrons. But doing so causes the pyruvate to break apart. And one of the carbons in the pyruvate leaves in the form of carbon dioxide. All we have left is now a 2-carbon compound, which is what enters the Krebs cycle. And it looks something like this. There's pyruvate over here. As soon as it comes into the mitochondria, it's oxidized by NAD+, which picks up 2 high energy electrons, and now we have NADH. NADH is pretty important to us because it's carrying 2 hydrogens and carrying high energy electrons. At the same time, we give off a CO2. This is the first place in our bodies that we give off carbon dioxide. It comes from the Krebs cycle inside the mitochondria. That's where all of our carbon dioxide, every bit of our carbon dioxide, comes from this location in the mitochondria at the beginning where the Krebs cycle starts. Now our remaining 2-carbon compound combines with a coenzyme and forms something called acetyl-CoA, and that then moves into the Krebs cycle. So what do you want to see here? This is the whole process of the Krebs cycle. And you notice there's blue pyramid, there's blue shapes over there with NADHs. Those 2 yellow dots represent the high energy electrons, and we keep making FADHs make high energy electrons. You'll notice that we're getting off, we're seeing carbon dioxide come off. Let me show you the bottom line in all this. This is, by the way, this is the Krebs cycle. And the Krebs cycle is something that, no one comes up to you and says, hey, so the 4th step of the Krebs cycle, you think your patient in bed 2 is giving off enough CO2 in the 4th step of the Krebs cycle? Never going to happen. There was a time that all of us that teach this could sit down and draw this out with all the reactions going on and explain it in great detail. And then the final exam was over and the test was over and we all forgot it. Basically what you have to understand is 2 things. The Krebs cycle is the source of capturing hydrogens and high energy electrons. It's also the source of carbon dioxide. Now, because no one's going to ask you any of these questions about here, this is what you need to remember. This is the Krebs cycle at the bottom line. What comes off the Krebs cycle? Well, we make a couple of ATPs in the Krebs cycle. We make 6 carbon dioxides. That's where all of our that represents all the carbon dioxide our bodies make. 6 carbon dioxides. It comes all from the Krebs cycle. What about those high energy electrons? The Krebs cycle, for every molecule of glucose, the 2 pyruvates that go through, you're going to end up capturing 8 NADHs. Each NADH is containing 2 high energy electrons. The FADH2s are each containing 2 high energy electrons. That's more important than a couple of ATPs. Because if you count those 2 ATPs with the 2 we made, the net 2 we got from glycolysis, that gives us about 30 kilocalories of energy out of 680. We're not there yet. What's more important are those high energy electrons captured over there in the oxidizing agents, the NADs and the FADs. Plus you picked up 2 more from glycolysis. So now we've got high energy electrons and we've got lots of hydrogens. What are we going to do with them? Well, we move further into the mitochondria, into the inner membrane of the mitochondria. The mitochondria has a double membrane, has an inner and an outer membrane. What you're seeing there on the slide is the inner membrane, and you have enzymes embedded in the inner membrane, and these are the electron transport chain enzymes. What will happen is that the NADHs with their high energy electrons will move up, will be drawn towards these enzymes, and they will give up their electrons. And the hydrogens will end up getting pumped across the membrane into the space between the inner and the outer membrane. Let me move on here and show you better. Okay. What we have here, okay, here's the inner membrane right there. Somewhere out here is the outer membrane of the mitochondria. And this area in here is the inner membrane space. Now, as our NADHs come up to these enzymes, they're going to release those electrons in here. Those electrons. And the energy from those electrons are going to be used to pump hydrogens into these spaces out here. So you have many more hydrogens in the inner membrane space than you do here in what we call the matrix. The only way to get those hydrogens out of there is for them to go through this device here called an ATP synthase. ATP synthase. As that goes around, it produces ATPs. I know it sounds strange, but that's exactly what happens. So let me show you here. These are the NADHs coming up, releasing their hydrogens and electrons. There goes the little white dots are the hydrogens. The energy from the electrons are used to pump them across. As they come back down, the only way they can get out is through these ATP synthases. And as the hydrogens go through there, that mechanically forces a phosphate onto ADP. So here's a little better animation than that. Here comes our first NADH right there. It is oxidized by the enzyme. It gives off its hydrogens, which are pumped across by the presence of the electrons. The electrons are transferred to the next enzyme, so it can pump more hydrogens across. And it's transferred to a third enzyme, so it can pump more hydrogens across. And then by that point, the electrons have lost their energy. The electrons have dropped down into the matrix. So now they're sort of free floating for a while. In the meantime, the hydrogens, which are up in the inner membrane space, the only way they want to reach equilibrium with the matrix, the only way they can do that is to come down through the ATP synthase. As they come down through the synthase, it causes the synthase to actually spin, and that takes inorganic phosphate and ADP and jams it together and creates our ATP for us. And notice how as we accumulate high levels of hydrogen in the inner membrane space, they gradually start coming down through. There they go. The hydrogens start coming down through there, and start forcing the third phosphate onto ADP. Now, once the hydrogens have come through here, they're in the matrix. What do we do with them? Well, it seems that when those electrons get done, you'll notice how the electrons are transferred from one enzyme to the second enzyme to the third enzyme, but then they end up down here in the matrix. Because they're no longer high energy electrons, they are low energy electrons. They drop down into the space there and they combine with hydrogens, which have come through. Hydrogens which have come through the ATP synthase now are combined with their electrons again. Now this is where oxygen finally comes into play for us. Atmospheric oxygen is split in half into elemental oxygen, grabs those electrons, grabs those hydrogens, and makes water. So we get our water at this point. We get our, underneath the green and blue bars is where the water formation takes place. The spent electrons are reunited with their hydrogens. Here come the electrons have one more shot. They pump some more hydrogens across. Now they're spent. They drop down into the matrix. They combine with the hydrogens which are already there that have been coming down through the synthase, and that forms water. Why do we want to do that? Because we got to get rid of those hydrogens. We want to keep high levels of hydrogens in the intermembrane space so they will continue to come down through here. If we reached equilibrium, we wouldn't make any more ATP. If we got to the point where the number of hydrogens here is equal to the number of hydrogens in the matrix, if the hydrogens in the intermembrane space up at the top is equivalent to what's in the matrix, then the hydrogens are not going to go through the ATP synthase and cause it to spin around, and the cell will die. This is why we have to have oxygen to keep pulling those hydrogens away. So we can always keep a high level of hydrogens in the intermembrane area. And this is pretty good as far as showing what's actually happening here. Notice how we have all these little pluses are the hydrogens here. These are all the electrons. We always want to have more electrons out here to grab hydrogens as they come through. When the hydrogens come through, they grab their electrons and are grabbed by elemental oxygen. We take atmospheric oxygen and split it in half. And this is not a bad animation either here. You can see we pump more hydrogens across. Our electrons pair up with the hydrogens that have come through, and that combines with atmospheric oxygen and gives us water. Without atmospheric oxygen, the whole process stops because all of our hydrogens in the matrix end up being the same concentration as what's in the intermembrane space. There's no movement. So we want to have that movement in here. And this is the process of oxidative phosphorylation. We get about 32 ATP from this. And if we combine that with the 2 we got from the Krebs cycle and the 2 that we got net from glycolysis, that gives us about 36 ATP. Okay, now, and there's another good animation of what's happening here. This is the ATP synthase. It is a little rotor that spins. They even figured out it spins clockwise. Doesn't when the hydrogens come through. As it spins, it mechanically forces the phosphate onto the ADP. So now, last slide, and then we're done with this unit. 3 terms: glycogenesis, the creation of glycogen. Genesis means creation. When we have excess glucose, we can store it in glycogen in our liver and in our skeletal muscles. If we have even more glucose, we will store it in adipose cells as fat. Another term, glycogenolysis, the lysis, the rupturing of glycogen to release glucose. We do this when our blood glucose levels are low, glycogenolysis. Third term, gluconeogenesis, the creation of new glucose. We break down fats, lipids, and we break down proteins. And we can convert the lipids and proteins into glucose and make new glucose so that our cells will still have energy. So, and those are the three terms. You need to remember those. And that is the end of, that's the end of Unit 2. So any questions on any of this? Okay, so I will get us out of here. And in lab this afternoon, we are going to go over the blood vessels. We're going over the blood vessels today. So if no questions, I'll get us out of here now.